Okay, we're going to get started. And our first speaker today is Eva Szabo. She's chief of the Lung and Upper Aerodigestive Cancer Research Group at the NCI Division of Cancer Prevention. She graduated from Yale University and then got her MD at Duke University. She did her internal medicine residency at Bellevue, New York Medical Center. And after completing a medical oncology fellowship, she joined NCI, and I've known her for many years. I'm sure you'll enjoy her lecture on non-small cell lung cancer. Eva.
Thank you, Terry. Everybody can hear me? Is this all two of you? No, sorry. Thank you for being here in person, and thank you all of you online. I was telling Terry that this gets weirder and weirder as you're talking to an empty room. But I've been told there are others listening in. So we're going to talk about non-small cell lung cancer. I think you've already had the small cell lecture. And I'm going to spend actually a fair amount of time about prevention, which is my day job, so to speak. So a little bit about lung cancer statistics. I've been doing this for many years, the many years that I've known Terry. And the number of new cases still continues to climb. It's over 220,000 a year. And almost as many deaths. For this year, the estimate is about 56,000. Now, there's no question that lung cancer is the leading cause of cancer deaths. It's greater than the next three causes of cancer deaths put together. However, the good news is that the death rate has been decreasing as we're decreasing tobacco, smoking. And even in women, you all are too young, but when I was a youngster, the tobacco ads were "You've come a long way, baby," Virginia Slims, celebrating that women have broken through, can smoke just as much as men. And then there you go. The lung cancer incidence went way up. It's now finally decreasing. Because lung cancer is the lung is a very deep internal organ, you don't find out that you have the disease until it's fairly advanced. So the 5-year survival is quite poor, 16%, 18% altogether, just a minor increase from 5 to 16% over the past 60 plus years. So a huge problem. Now the risk factors are well known. Yeah, here we go. 85% or so is due to tobacco. That includes passive smoking. So if you grow up in a family where there's a lot of smoke or your spouse smokes, that's bad. It's a field effect. So if you've had a prior aerodigestive malignancy, head and neck cancer, for instance, you're at increased risk of having a secondary lung cancer, chronic obstructive pulmonary disease, COPD. But here's really the picture. So this is a picture of somebody with a lung cancer. Here's all the fibrotic changes that are, and it's hard to see, but this is some COPD there. And there's the pack of cigarettes. Okay, and there you have lung cancer in a nutshell. There are other exposures that are also important. Asbestos, well known. Obviously, we've been trying to get rid of that over the many years. Indoor radon, we check our houses, right? Our basements when we're lucky enough to buy a house. And then a number of exposures that people get as part of their jobs, chromium, nickel, et cetera, from various industries. Inorganic arsenic, which is in groundwater, not so much in most parts of the US, but in other parts of the world. Genetics has been a little bit hard to study because when you live with smokers, you also get their genes if you're a child. But nevertheless, there are some genetic predispositions. Very rare germline mutations in the epidermal growth factor receptor gene, the T790M mutation. And I'll be talking about this a little bit because this is an important target for cancer treatment. There are various nicotinic acetylcholine receptor subunits that are associated with increased lung cancer risk. And interestingly enough, one of these is also a susceptibility locus for COPD, helping to put this together why COPD is a risk factor, one of the reasons why COPD may be a risk factor for lung cancer. This is by no means an all-inclusive overview. I'm just giving you sort of the highlights. So non-small cell lung cancer is a basket term. It really includes several different histologies, the most important one being adenocarcinoma. And it's got specific patterns of how it looks under the microscope. These tend to be peripheral nodules that obviously spread. And this is the most common type of non-small cell lung cancer. And it includes the non-smoking associated lung cancer, but most adenocarcinomas are still due to tobacco exposure. About 20% are squamous cells. These tend to be more central tumors, different appearance under the microscope, and more heavily associated with tobacco exposure. Somewhat less common are the large cells, which also tend to be more peripheral and not as clearly associated with tobacco, and then a smattering of other rare types of histologies. But we tend, at least in the past, we tended to lump everything together. It is not the small cell that you learned about a few weeks ago. And here's, of course, small cell. You learned about that. So the development of lung cancer it's a process, and it's a process that therefore allows us many opportunities for intervention, although the bulk of what we do in medical oncology is right here, early to late cancer, which once we find it, we cut it out, we radiate it, we give various systemic treatments. I will be talking about the much longer phase from the time of initiation where the exposures to tobacco, for instance, come in and damage the respiratory and alveolar epithelium through the gradual changes, dysplastic changes, until you have the development of a locally invasive and ultimately metastatic cancer. So the treatment strategies for lung cancer are very similar to other types of cancers. They're very anatomically based. Early stage, it's surgery, you take it out. If you have more local involvement, including some lymph nodes, either if you can still resect, then you generally follow up with adjuvant chemotherapy, and that has been actually shown to improve survival. So that's standard for stage 2 and stage 3A. Once you get past just the very localized tumors and you start to have lymph nodes in the mediastinum, then you're generally talking about combined modality therapy. So that's usually chemoradiation, and sometimes you even add surgery to that. But once you get to the more extended involvement, either what used to be called 3B wet, in other words, pleural effusion, or which is really systemic spread, or any kind of distant metastatic disease, then you're talking about systemic therapy. You can use radiation as needed for local control. Sometimes you will, if you only have one isolated metastasis, for instance, in the brain, which is a privileged site, you may cut it out. But for the most part, you're talking about systemic treatment. And of course, that's the world that I live in as a medical oncologist. And small cell lung cancer, you heard about. So what's changed is that we now understand a lot more about the genesis of various types of lung cancers. And we now pay attention to the histology as well. So it turns out that there are a number of different driver mutations that are present in adenocarcinomas. And in fact, we can ascribe various drivers to about 2/3 of all adenocarcinomas. And for some of them, not that one, for some of them, they actually serve as a basis for targeted therapy treatment. And so this really started out with the epidermal growth factor receptor, which is a very important component in cell signaling. Turns out that about 10 to 15% of adenocarcinomas have EGFR mutations. And if you have an EGFR mutation, then there are various drugs, erlotinib, afatinib, gefitinib, a series of drugs that will give you a very hard, these are oral tyrosine kinase inhibitors, and they interfere with EGFR signaling, and they are associated with very substantial and prolonged response rates. So in contrast to chemo, which about 20 to 30% of people will respond to, and usually for about 5 or 6 months, if you have an EGFR mutation, the response rate is 3 times as large, and PFS, progression-free survival, is doubled. Okay, and median survival is also about doubled. This is a minority of people. Okay, but this is a very important treatment, and this is standard of care. If you have somebody with advanced non-small cell lung cancer, you will check, especially adenocarcinomas, you will check for EGFR mutations. We now know a lot more about the mechanisms of resistance. We have second-line drugs. Osimertinib is one that deals with a very specific mutation that in the EGFR receptor, which occurs in about 50% of people who progress after these first-line drugs. It's the T790M mutation. And again, you can get extended progression-free survival, although ultimately everybody does progress and die. And interestingly enough, this T790M mutation is the one that I mentioned earlier, which is a familial form of non-small cell lung cancer. There are other molecular abnormalities which are very important and which we check for when somebody has stage 4 disease. The EML4 ALK fusion gene occurs in about 5% of non-small cell lung cancers, again, mainly adenocarcinoma, and just like EGFR, mainly in never smokers, okay? And there are now 4 drugs that are actually approved for treatment, very similar to the EGFR story, about 60%, 50 to 60% response rate, and about 8 to 14 months duration of response. So if you are unlucky enough to have lung cancer, but lucky enough to have one of these molecular abnormalities, your prognosis is much better. There's more options. And of course, after progressing on these various oral tyrosine kinase inhibitors, chemotherapy still remains an option. ROS1 is another abnormality, similar story, perhaps better duration of response using crizotinib, which is the same drug that we use for ALK translocations. BRAF, FDA recently approved, this is much less common, about 2% have the V600E mutations in BRAF, 2% of non-small cells. This is the same mutation you find in melanomas, and this is the same regimen that gives very similar results in melanoma and in non-small cell. RET, MET4 exon skipping, the reason I'm really putting this on is to show you that these are now all important because they serve as a basis for treatment, but rare mutations. And so you can imagine the complexity, because how do you know that somebody has a mutation? You have to have tissue. And so now we get tissue on everybody, and we send it for molecular analysis, and it's expensive and difficult, and some people you can't get enough tissue. So you can understand that the science of how we, or the art of how we do treatment for lung cancer has changed, and it's complex. When it comes to squamous cell, we don't have the same options. So all of these things that I showed are primarily in but not exclusively, in adenocarcinomas. You know, they're the fibroblast growth factor receptor 1 amplifications, which there are inhibitors in development, but since 2010, nothing has come on the market. So this is not the same story of driver mutations that you can target. This is another abnormality, again, now in a very small percentage of squamous cell carcinomas. In vitro, we have drugs that can treat it, but since 2011, this has not come on the market. So therefore, this has not been as targetable as what I described in adenocarcinomas. What is even more new, if I can put it that way, than targeted therapy, and non-small cell lung cancer is the poster child for targeted therapy, it's also the poster child for immunotherapy, and that's what you see in the news every day. The program death ligand, this all has to do with T-cell activation. I am not going to do justice to discussing immunotherapy, but the bottom line is that the goal here is to undo the T-cell limitation of activity or T-cell inactivation, and to reactivate the T-cells, which are often blocked in the setting of cancer. And so it's a ligand. This is just the beginning of immunotherapy. The ligand for this receptor, which inhibits T-cell activation, is PD-L1. And there are now a number of antibodies approved, some of which target PD-1, the receptor, versus PD-L1, the ligand. And so here's just an example of why this is so exciting. So we now have approvals for both second-line treatment and frontline treatment with several different antibodies, 3 for second line, 1 for frontline. And the reason that this is really key is, if you look here, is that there seems to be a plateau to the survival curve. So people who respond tend to respond for a long time, sometimes for years. And that is a game changer for anybody who's ever treated lung cancer patients with chemotherapy or even targeted therapy. They will stop responding, and usually that's less than a year. But some of these responses have been extremely long-term, and so people are very excited about the possibility that perhaps some people can actually be cured after having metastatic lung cancer. Let me just say that it's a small percentage of people who have these kinds of outstanding responses, but that's something new. So for instance, for frontline treatment, if you have high expression of the ligand, which is somewhat predictive of response, not exclusively predictive of response, then the overall progression-free survival is almost doubled. But again, what's really exciting is that people who respond tend to stay responding for a long period of time. So that's, in a small nutshell, the landscape for treatment. So as exciting as it is, people don't really get cured. And I will never walk into a patient's room and say, well, I'm going to give you immunotherapy and I think you have a chance at a cure. You know, I'm hoping that that is going to turn out to be the case eventually, but essentially if you have stage 4 disease, that is a death sentence. So what can we do? So obviously we're going to continue to work on the therapeutics, but I'm going to now turn to prevention and early detection. So smoking, important to know. Obviously it's the risk factor, but the converse question is, so if you smoke, you increase your chances of lung cancer. If you stop smoking, do you decrease your chances? It's not, the answer is not as simple as you would think. So these are the long-term results of a study. It was actually a study of COPD called the Lung Health Study, 15-year, 14.5-year follow-up. And what you see here is the death rate, okay, per 100,000 from lung cancer, from other diseases, okay. If you continue to smoke, it's 3.5 per, sorry, not 100,000, per 1,000. If you quit, okay, then it's about half of that. And if you intermittently quit, it was somewhere in the middle. So if you've quit for 15 years, your death rate from lung cancer is really decreased. If I had shown you the data from the 5-year follow-up, there'd be no difference. And the reason for this is because lung cancer, tobacco damages the epithelium. There's genetic damage. There's slow turnover. And so if you continue to smoke, your risk continues to go up. if you stop smoking, you live with the risk that you had. So if you continue to smoke, there's your rate of lung cancer. If you stop smoking, it plateaus. Okay, so should you quit smoking? Absolutely. The sooner, the better. But if you quit smoking today and you've been smoking for 30 years, don't think that your risk of lung cancer goes down quickly. It doesn't. It goes down for cardiovascular disease much faster than for lung cancer. Okay, so let's talk a little bit about prevention and really about what we call, because you will continue to be at risk, about cancer chemoprevention. This was just defined many years ago by Michael Sporn, who had been here at NCI at the time, as the use of natural or synthetic agents to suppress or reverse the process of carcinogenesis. As I tried to tell you, you get all this damage to the respiratory epithelium, and that progresses over time. So with chemoprevention, you try to undo the damage. So lesions that are partially progressed, that are pre-neoplastic, you want to regress those. You want to prevent new lesions from getting to the point that they're pre-neoplastic, and to suppress those that are neoplastic already. And so the rationale, therefore, for trying to prevent lung cancer is because no matter what I told you and how exciting immunotherapy and targeted therapy is, the lung cancer survival still is awful and unacceptable, okay? And we know from other diseases that actually cancer is preventable. So I don't know whether you've had the breast cancer lecture yet and whether you've heard about the tamoxifen and raloxifene trials, but these are trials in women who were at risk but did not have breast cancer yet, and you can decrease the risk of subsequent breast cancer by 50%. So, and of course, you can model this in animals in many different ways. So it's possible. Question is how you do it. Knowing that there is this long preclinical phase with these increasing histologic and molecular abnormalities, primarily due to tobacco exposure, means that there's an identifiable population that you could study and hopefully intervene in. So when do we want to intervene? Well, it depends, or why. It depends on the efficacy. If we can intervene when you have metastatic disease, that's when you want to do it, right? Those are the people who need it. But we know that we do better with early stage cancer than with late stage cancer. We can cure people by surgery and adjuvant chemotherapy. We don't cure them with metastatic disease. So we presume that the precursor, the pre-neoplastic lesions, are more curable than the invasive ones because they tend to be less molecularly complex. That's a hypothesis, okay? Can we prevent the carcinogen-induced DNA damage in the first place? Probably, but you need to start that when you're born, okay, or probably in utero. And we need to be aware of the fact that I told you about the various driver mutations, many of which are in never smokers, but some of which occur in smokers. And they're different pathways to getting something that looks similar under the microscope. So you need to have interventions that can address these multiple pathways of carcinogenesis. So that's one issue. You need efficacy, which we think earlier is better than later. You need to look at toxicity, because if you're talking about people who have metastatic disease, you can get away with bone marrow transplants and things, chemotherapy regimens that have very high rates of toxicity, including having people wind up in the ICU not infrequently. That's not the case in people who feel healthy, who may never develop cancer, right? So the toxicity has to be acceptable both short-term and long-term. You can't increase the risk of other abnormalities like cardiovascular disease, okay? Third consideration is the size of the target population. There are 44 million current smokers and about as many former smokers in the US. It's not, obviously they're not going to all develop lung cancer in their lifetime, about 1 in 8, never mind right now. So you have to be able to figure out who are the highest risk smokers. And of course, we can identify the non-smokers, the never smokers, who are at risk because It's a substantial number of people, but it's still a relatively rare population. So these are all issues, and cost is always an issue. So how do we identify agents? Well, we know the mechanism, and I already told you there are many different mechanisms for lung cancer. We can do better. HPV vaccine and cervical cancer. That's not to belittle how difficult it was to get to this point, but you understand the pathogenesis. You have a vaccine. You could prevent essentially almost all cervical cancer. So with lung cancer, that's a lot more difficult. So we turn to preclinical models. We look at epidemiology, and I'll give you some examples of all of this. And we look at secondary endpoints from clinical trials. This is how we found tamoxifen and raloxifene for breast cancer. Okay, so target selection, cohort selection, risk, benefit. What are the endpoints that we look at? And to design the clinical trials the right way. Those are the major issues. That's what I really spend my days worrying about. Okay, so I'll tell you a couple of stories of things that we've tried to develop. One of them, and not just we at NCI, but others in the field, and one of them is the inflammation story. There's a lot of data going back to the 70s, showing a role for steroids in cancer prevention. This is primarily in animal models. This moved on to lung models, oral and inhaled steroids, the work of the late, I would say, great Lee Wattenberg, who really was a mentor. The epidemiology has been not as strong, but most steroid use has been in the setting of asthma, which is not the high-risk condition. COPD is the high-risk condition. And there's some studies suggesting that those smokers with COPD who use steroids have a lower risk of cancer than those who did not. This is an example of an animal model. This is a vinyl carbamate. If you give vinyl carbamate to mice, you get about 80% decrease in the number of tumors, okay, depending on the amount in the diet. This is budesonide, which is one type of steroid. And not only do you get fewer tumors, but instead of having carcinomas, you really revert them back to adenomas. So you prevent the progression, decrease numbers, not as advanced histologically. So how do you bring this to people? Well, in the bronchial epithelium, we know the progression to squamous cell carcinoma. It goes through varying grades of dysplasia, of abnormalities, until you get to carcinoma in situ and then invasive carcinoma. And these lesions can be identified on bronchoscopy, and they tend to progress to more severe lesions if you follow them over time. Not all lesions progress. In fact, the majority don't. It's hard to know which ones will, but the ones that are mild, like the metaplasias, really will regress as much of the most of the time, and only infrequently progress to carcinoma in situ, whereas the severe dysplasias will still regress about half the time, but a third of the time they go on to progress, and the carcinomas in situ tend to progress in almost half the patients. So the more severe the lesion, the more likely, if you go back to look, that you will actually get to a cancer. This is an interesting study that showed that in people with low and high grade bronchial lesions, about a third developed invasive cancer. However, only about 40% of those were at the site of that abnormality. The other 60% were someplace else, either along the bronchial epithelium or even in the periphery of the lung as adenocarcinomas. So this tells you that these lesions are both precursors, because some of them will go on to develop cancer, but they're also the risk markers for an abnormal field because of tobacco exposure. So this is what we have focused on, are these lesions, because you can go back, as hard as it is, you can go back with repeated bronchoscopies, looking in, biopsying, and then asking, Do these lesions get better? Are there new lesions? So we took inhaled budesonide, which is what the mouse study showed, was the data that I showed, took 112 smokers who had dysplasia in the bronchial epithelium. They also underwent helical CT. They were given budesonide or placebo inhaled for 6 months. And then we went back, we bronched them, and we repeated CTs and to see how well they did. This is work of Stephen Lamb in British Columbia Cancer Agency, who's one of the few who can do these types of studies. The results, the data from that study, are that we really did not have an effect on bronchial dysplasia, although the CT detected lung nodules actually tended to resolve more frequently in those who had budesonide versus those who did not. Now, why would this be? Well, the animal model that we used, which was really the only type of model available at the time, gave mice little adenomas, not squamous cell. And what we were looking at in people, in retrospect, were the bronchial atypias, which give rise to squamous cell carcinomas, not the adenos. What you find in the periphery of the lung, the CT-detected lung nodules, are more likely to give rise to adenocarcinomas. So we figured this out retrospectively and decided to look in the periphery with the advent of low-dose CT scans. And so this is the sort of the next study in this process, where we took people who were undergoing CT screening. This is the work of Giulia Veronesi and colleagues in Milan at the European Institute of Oncology. They had a screening cohort, and we randomized people to get inhaled budesonide or placebo for the year in between their repeated CT screens. Now, this was the first time we've ever actually looked consecutively in the peripheral lung, and we found many different types of nodules. And obviously, we ruled out that people had actual lung cancer. So we were only looking at what's called the indeterminate nodules, which you would follow but not take out because they're small. So the solid nodules, that had been there, stayed there, didn't change in size. The non-solid nodules, however, did change in size. They got smaller. And and this was not statistically significant because this was only about 30% of the people who had these kinds of nodules. But as we followed them over time, they got a year of inhaled budesonide, and then they continued to get screenings. As we followed them over time, those who had budesonide still continued to get a little bit smaller, while those who had placebo did not. So not a home run, but at least telling us how maybe we can do these studies better. So what are these nodules that I'm talking about? They look like this. They're these ground glass, not fully solid nodules. Under the microscope, some of them look like this. This is what's called atypical adenomatous hyperplasia, AAH. It is about a quarter to half of these kinds of nodules will be AAHs. Others could be carcinomas or some fibrosis or various other pathologies. And it turns out that the belief is that these nodules, some of them are actually precursors. So this is one study that followed patients over time with these ground glass opacities. And basically about 1% of them will become invasive over time. Some of them will become what's considered non-invasive but cancers, adenocarcinoma in situ, minimally invasive adenocarcinoma. So these nodules, some of them are precursors. I'll give you sort of a real-life example, and it shows the complexity. This is actually a friend of the family who also became one of our patients. Woman who developed chest pain, went to the ER. They thought that maybe she had a pulmonary embolus. She didn't. They did a CT scan, and they found this. This is a ground glass opacity. Okay, this is April of 2004. as is the standard of care. She was told to come back in 3 months. Didn't really change a whole lot. She was told to come back in a year, and life takes you in various ways, so she didn't. Then wound up in the ER again 6 years later. And now, again, chest pain, didn't turn out to be anything. CT scan showed this. This is now a little bit bigger than this. So she went back a year later yet again, and now it's more solid. And so this turned out to be an invasive adenocarcinoma with areas of atypical adenomatous hyperplasia. So this was clearly the precursor. Here's the cancer, 7 years, okay? So if we had done a study using her as one of the participants, we would have gone from this to this, somewhere in between. We would have probably gone from this to something that looks just like it in a year. So that's part of the difficulty of doing the studies. So we've gone back to ask the question a little bit more again. If you have non-solid nodules, what's the risk of developing lung cancer? These are data from the National Lung Screening Trial, which was done, coordinated from the National Cancer Institute. And the bottom line is that we looked at people who had these small nodules, ground glass versus other type of abnormalities, solid abnormalities, and 5 or more years out, they had about a 3-fold increased risk of lung cancer. So some of these ground glass nodules are lung cancer precursors. Others are not. So what are we doing now? So now we're looking at aspirin. Okay, so to continue the story of inflammation. And the reason we've turned to aspirin from inhaled drugs is because, number one, it gets every place more evenly because it's systemic. But it's really from these studies by Peter Rothwell and colleagues. And it's a whole series of studies looking at the long-term outcomes in people who were randomized to receive aspirin or placebo for cardiovascular disease, many of whom, by the way, smoked. These were old studies. And when you look at the long-term outcomes in terms of cancer deaths, 5 years or more out, you see that the curves diverge, and you get about a 20% decreased risk of cancer death 5 years or more out. And the longer, the better. And if you look at various types of cancer, this little teeny one here is lung cancer. Actually, this curve diverges within about 2 years. Again, this is cancer death. And this is lung adenocarcinoma, where you have about a 30% decrease in cancer death by 5 years. So we went back with Dr. Veronesi and colleagues in Milan to ask if we take people now with these pre-malignant lesions, the AAHs, the ground glass opacities, so not the solid nodules. We screen them because they're part of a screening program. We give them aspirin for a year. Can we get a better outcome than with inhaled budesonide? And this is still ongoing. And we also decided to look a little bit more molecularly to see what is the effect of aspirin short term. And so in this study, what we're doing is we're doing nasal swabs. So there's a whole series of data showing that there are abnormalities that you can detect by gene expression, various signatures that are associated with smoking versus non-smoking, those who have cancer versus somewhere else in the lung versus those who don't, and that these signatures are similar in the nasal epithelium as they are in the bronchial epithelium. So we're looking at the short-term exposures to aspirin to complement the CT nodule study. I'm going to tell you one other very short story because, again, it's not a drug that we're ready to tell people you can use, but because it shows how we're changing how we do these studies. So this is myo-inositol, which is actually a glucose isomer, source of several signaling molecules. It's actually found in rice and baby's milk and various grains and so on. It's a food constituent. It's actually on the list of what the US FDA calls GRAS, generally regarded as safe. which I think for prevention is very important. And so there have been many studies, again, the work of Wattenberg and Steve Hecht, that showed that myo-inositol in animals can inhibit carcinogen-induced tumors, both in smoking-exposed animals as well as in carcinogen animals. If you combine it with steroids, you really increase the efficacy. So we decided to go with our colleague Stephen Lamb in Canada to do a phase 1 study, figure out what can we do in people, and found a tolerable dose of 18 grams per day. This was an uncontrolled study because we wanted to know the dose that is correct, but people underwent bronchoscopy, and compared to historical control, which is a dangerous thing to do, you can do it as to push you in the right direction, but you can't assume that this is the correct answer. So compared to the historical controls, we found a very high rate of regression of bronchial dysplasia. We found the dose, preliminary data. We were lucky enough to have bronchial brushings from these airways, and Avi Spira at Boston University looked at gene expression in these bronchial airways and showed through a long series of elegant studies that the PI3 kinase pathway is activated. This is now in the normal bronchial epithelium in smokers with dysplasia to a much greater degree than healthy smokers or non-smokers. And myo-inositol, this is a small number of patients, inhibited this PI3 kinase activation in this normal bronchial epithelium. So this opens up the possibility, two possibilities. One, how important is PI3 kinase? Well, actually, PI3 kinase is frequently activated in squamous cell carcinomas, head and neck, lung as well. So does that identify the truly high-risk smokers? Not sure yet. But it also gives us this new clinical trials model where we can do these shorter studies and look at different outcomes, not the bronchial dysplasia, but actually looking at gene expression signatures. So that's how we're trying to move forward. So to look at that, we did the phase 2b study, which means that we randomized people to myo-inositol placebo. Everybody got a bronchoscopy pre and post-treatment, and also gene expression signature was assessed from the normal bronchial brushings. Primary endpoint was not nearly as promising as was the small phase 1 study, which is why I cautioned you not to believe that. So there was a slightly higher rate of complete regression with myo-inositol, but also of progression with myo-inositol in all subjects. So it really came out a wash. So this was really a negative phase 2b study. With increasing abnormalities, there's increasing Ki-67, so proliferation. Placebo versus myo-inositol, not statistically significant, but definitely a trend towards more inhibition of proliferation with myo-inositol. And downstream of PI3 kinase, AKT pathway, the activation was decreased in the complete responders, but not in the others. Similar data to the phase 1 study, but doesn't help us figure out who's going to respond and why. So we're continuing to look at all this wealth of information to try to figure out whether we can identify who should get myo-inositol. It's obviously not for all smokers. Very heterogeneous response, and we need to do better. All right, so those are examples of how we are doing studies. There is no approved strategy for lung cancer prevention right now, other than stop smoking, and that will help you years down the line. How about early detection? So very quickly, Nothing in this life is free, okay? And so the issues with lung cancer screening are also complex. And that's because you're looking at various time points, and our screening is subject to various biases, something called lead time bias, where because you're screening, you're finding the diagnosis at an earlier time, but you're not actually postponing the time of death. Survival only appears longer. There's something called length bias. You're more likely to find the indolent disease as opposed to which has a longer preclinical phase. It's well known with mammography that the people who have really bad disease often pop up in between the mammograms. Okay? There's something called overdiagnosis. You identify actually clinically unimportant lesions. They look like cancer, but they would have never been diagnosed, and they never would have caused the person's deaths. And of course, anytime you do anything to people, there's morbidity, there's mortality, and there's a cost. This is the PLCO, chest X-ray randomized trial. Okay, can doing chest X-rays reduce lung cancer mortality? 154,000 plus participants, just one chest X-ray versus usual care, 4 screens, 13-year follow-up, no change to mortality. Okay, so just getting a chest X-ray isn't going to help you. How about CT? So CT was developed in the 90s, and so we have the National Lung Screening Trial, the NLST, which is really the biggest randomized controlled trial of CT screening. 53,000 smokers, current and former, at least 30 pack years, had to have quit within 15 years. And this was a randomization between helical CT and chest X-ray. And so even before we knew that chest X-ray was definitely not helpful. Okay. The results were that there were about a quarter of people had a positive test. Okay. 354 lung cancers. Chest X-ray, many fewer positive tests, 442 lung cancer deaths. Overall, a 20% reduction in lung cancer mortality. Highly significant. There was even a significant reduction in all-cause mortality. And this is just a picture showing cumulative number of lung cancer cases. There's CT versus chest X-ray. But if you're looking at deaths, it's the chest X-ray that's on top. That's what you want to see. You detect more and you have fewer deaths. So, good. So, this is now being worked on to make it person-friendly and to optimize, because that's screening a lot of people to get a 20% decrease in mortality. So this is example of one study that looked at various parameters, age, sex, family history, emphysema, types of nodules, and could tell you the risk of having a lung cancer based on that first screening CT. Okay, this is work from Canada again. So this would allow us to identify those at highest risk for subsequent screenings, as well as potentially for future phase 3 chemoprevention trials. So what we want to do is we want to identify those who are most likely to actually have lung cancer subsequently and eventually die. Those are the people we want to intervene in. Okay, just again, screening should be a whole different lecture. So let me just sum up then. There's been huge progress since the time I entered and came to NCI, which you didn't mention the year, Terry, in understanding lung cancer, the genesis of lung cancer in a molecular way. Precision medicine is applicable now to a significant but still small subset of advanced stage lung cancer patients. There's clearly increased survival in this subgroup. We're now in the early days of immunotherapy, but already there's been very significant prolongation of survival, again, in a small subset of patients. And we can decrease lung cancer mortality with early detection with helical CT. So work is ongoing, and it's it's actually there's an enthusiasm that was not there 20 years ago, but there's still a lot of work to be done. And with that, I will go back to my prevention roots. Ounce of prevention is worth a pound of cure, and just to thank a number of people who've actually done the work. And I'm happy to answer questions. Thank you. Yes, sir. It's oral. It's a powder. Right. Good question. We have no idea. Okay. And so that's the kind of study that you would want to do. Well, you would want to do it in two settings. One is that if you really think that there is a subpopulation who are truly responding and you want to understand the mechanisms, or if you think that there's a problem there that you can fix to help the efficacy, then you would want to do the studies. Until we get a strong enough signal that this really is a good chemopreventive agent, I'm not sure it's worth looking into it. But it's a very good question as to whether that's what's going on. No. Not for lung cancer prevention. You know, there are guidelines for using aspirin for cardiovascular disease, and for those who are at risk for cardiovascular disease, which smokers are at increased risk, but you know, with to a specified amount, to actually use aspirin for colon cancer prevention. But there's, and that's actually in the guidelines now, but it's, you know, it's for ages 50 to 59 or something, and consider it, you know, in the next age group. There's really not enough data, and the history of cancer prevention is littered with good intentions of studies, you know, the beta carotene studies that I didn't talk about, where the outcome was worse with the intervention as opposed to better. So until you really have good clinical data, I would not do that unless she's at high cardiovascular risk. But tell her to stop smoking. All right. Thank you very much. I'm going to turn this off.
Lots of hope.
There's hope. There's always hope.
Okay. All right, so our next speaker's here, and we'll get started. So Len Neckers, he's a senior investigator in the urologic oncology branch. He got his Ph.D. from the University of Connecticut, did postdoctoral training at NIH, and joined the NCI in 1981. He's chief of the tumor cell biology section, and he's going to talk to us about targeting molecular chaperones in cancer. Lessons learned from HSP90. Len, welcome.
Thank you, Terry. It's a pleasure to talk to you guys about this. This is a little bit of a change of topic from the last speaker. But I think it provides some interesting examples of how novel targets for cancer treatment may at first appear to be surprising targets, and then perhaps the expectation is to have great results, and when the great clinical data don't come in, one needs to start looking for explanations. And usually the explanation is that things are more complicated than we thought, and that's what I hope to show you here. But it shouldn't stop us from continuing to explore, to understand what the deficiencies are. Some of the things we know now we didn't know 20 years ago when we started developing these agents. And I think that the future suggests that chaperone inhibitors may still be useful, both for cancer and for other diseases if used appropriately. So with that in mind, I'm going to talk to you about a particular chaperone that I've been working on since the early 1990s, HSP90. Although we didn't so much appreciate it then, HSP90 and other molecular chaperones are just part of a group of effector proteins that make up the proteostasis network, or PN. And this network, which includes the proteasome, autophagy, directed proteins, the unfolded protein response in the endoplasmic reticulum, mitophagy, etc., are all means that the cell and the organism uses to cope with environmental stress, which leads to damage of proteins in the cell. This has to be dealt with. If it's not dealt with, this accumulation of misfolded proteins is going to kill the cell. So looked at from that perspective, I suppose it's not surprising that cancer cells, which, as I'll show you, suffer from all sorts of environmental stress pretty much all the time, have grown very dependent not only on HSP90, but on the proteostasis network in general. And there are very few, but some examples of drugs that target this network. HSP90 inhibitors have been in the clinic since the late 1990s. A number of proteasome inhibitors have been in the clinic for almost as long, and in fact, 2 of these have been approved by the FDA for certain types of cancers. So understanding that the proteostasis network is a target is appreciating the fact that the infrastructure which allows cancer cells to survive and multiply may in fact be a useful target for treatment. So in normal cells, as I just mentioned, HSP90 in conjunction with other members of the PN helps to maintain protein homeostasis or proteostasis. In cancer cells, this becomes a much more complicated and more difficult task for both HSP90 and the proteostasis network in general, because there are many mutated, overexpressed, or activated oncoproteins in these cells that are inherently unstable, don't fold easily, and need to either be chaperoned through multiple rounds of chaperoning to allow them to fold, or if the cell senses that they're never going to fold, then to target them for degradation in the proteasome in particular, so that they don't accumulate and cause toxicity. So in terms of HSP90, it's been known for a long time. These are data taken from a paper in 2013, but yeast experiments going back to the early 1990s confirmed that yeasts that are growing at optimal temperature and growth conditions have maybe 10-fold excess the amount of HSP90 that they need in order for survival and for growth. And that's just shown here in this graph, which looks at expression level of HSP90 on the x-axis and relative growth rate on the y-axis. And you can see that relative growth rate in these yeasts is not impacted until levels of HSP90 are reduced by upwards of 90%. So these are under optimal conditions. If you look at this HSP90 point mutation in the red box, this mutation weakens the activity of HSP90. It doesn't destroy it. And as you see here in this yeast dilution assay where yeast cultures are spotted at a number of serial dilutions going from left to right, and then one observes their growth just by looking at how dense these white circles are. At 30 degrees, which is the optimal temperature for yeast, this function-compromising mutation has no impact in yeast growth. But if you switch the yeast to 16 degrees, which is definitely not optimal for their growth, while they're not affected if they express wild-type HSP90 or these other two mutants not in the red box, but the mutant in the red box is now no longer able to support the growth and viability of these yeast. Another way of looking at this is by expression strength. Now you can do a similar experiment to comparing the 30 degrees to 16 degrees. You can do it all at 30 degrees, again, optimal temperature, but you can vary the expression of HSP90 like I showed in the original slide panel on the left of the first slide. So you can see if you look at the wild type, which is in the green box up at the top, pretty much you can reduce HSP90 expression to less than 10% of normal. And the yellow color shows that the yeast are able to tolerate that and are growing fine. If you look at the W585T mutation, which is shown in the red box, you can see that at 30 degrees at optimal concentration, like I showed you in the previous slide, the yeast are happy. But as you begin to decrease expression, even though growth conditions are optimal, it begins to not work and to not support yeast growth. So the reason I'm showing you all this is that the bottom line is that in yeast as well as in human cells, there's generally an excess, a large excess of HSP90. You can reduce it a lot and have no obvious effects. But if you stress the yeast, either by giving them an HSP90 that is not completely active, or if you put them at a temperature that they don't like, they now become very dependent on optimal expression. And one can imagine a similar situation for cancer cells. So the green line is showing what would happen to a normal cell if you reduce the HSP90. You'll get to a point where it's going to have an effect because HSP90 is essential for growth. But normal cells don't need all of the HSP90 that they have. In normal cells and tissues, HSP90 makes up somewhere around 3% of the total protein of the cell. So it's quite a lot. It's in micromolar concentration in cells. And they can get by with a lot less. But if you stress the cell, or instead, if you look at a cancer cell, which as I'll show you in a minute, is essentially stressed all the time, then they can tolerate only a little reduction in HSP90 levels before you see significant effects on growth and viability, or fitness, as shown here on the y-axis. Now, it's not exactly correct to equate expression with inhibition, but an analogy can be made suggesting that you can inhibit HSP90 to a sufficient degree where you're going to see an effect in cancer cells while you see minimal impact on normal cells. And over the years in published papers, this is in fact what we see. So cancer cells in general tend to be more sensitive to pharmacologic inhibition of HSP90 compared to normal cells. And a likely reason for this is that HSP90 is one of the main response mechanisms that the cell and the organism has to environmental stresses, some of which that are common to cancer are shown here. On the left-hand side, cancers usually outgrow their blood supply, and so nutrient stress is a common factor. There's proteotoxic stress because there's usually a lot of reactive oxygen in cancer cells. They're hypoxic, so they're not getting enough oxygen to have the mitochondria function at optimal activity. And many, if not most, cancers are aneuploid and are characterized by extreme genetic instability. This leads in turn to the synthesis of mutated proteins that are not able to fold and must be dealt with by the cell, either to chaperone them until they're folded or to target them for degradation. And HSP90 is involved in both of those processes. So all of these stresses impinge on HSP90, and HSP90 in turn controls the fate of a host of client proteins that are signaling proteins, for example, the one shown on the right-hand part of the slide, that mediate a lot of the processes important for cancer cell growth, survival, metastasis, etc. The gray boxes describe what are now accepted as the hallmarks of a cancer cell that distinguish cancer from normal. And you can see that all of the hallmarks that are shown here, there are client proteins that depend on HSP90 that are important for each of them. So that you would think that by targeting HSP90, you can have a major impact on the viability of cancer cells in vitro and in vivo. And a correlate of this would be to suppose that if you stage cancers by the amount of HSP90 they have, the more HSP90 expressed in a cancer, the more aggressive the cancer might be and the worse the survival or the prognosis of the patient would be. And this, in fact, has been shown by looking at tumor tissue that's taken from patients for a number of different kinds of cancers, including non-small cell lung cancer, which I'm not going to show you today, but also in breast cancer, which I'm showing you on this slide, where if you stratify the tumor samples from patients with triple negative breast cancer and make a cutoff where above that is arbitrarily called high HSP90 and below that is low HSP90, you can see that in tumors where HSP90 is upregulated significantly compared to the remainder of the population, percent survival is markedly reduced in these patients. So expression level of HSP90 is a negative prognostic indicator of cancer aggressiveness and survival. It's a prognostic indicator of aggressiveness. It's a negative prognostic indicator of survival. So our idea was that inhibiting HSP90, like knocking down expression of HSP90 in simple systems like yeast, should compromise cancer cell robustness. And this, in fact, turns out to be the case. This is an experiment we published a long time ago now that shows that this prototypical HSP90 inhibitor, 17-AG, which is the first in human HSP90 inhibitor, is very effective as a radiosensitizing agent, where looking here at a couple of lines, HeLa cells and CHA cells, and you can see the doses of irradiation on the x-axis and the surviving fraction on the y-axis, and the y-axis, make note, is in a log scale. So you can see that at this dose of radiation, the HeLa cells and the CHA cells, which are both lines derived from cervical cancer, are sensitive, but certainly there's a significant fraction of cells that remain after irradiation. If you add in low doses, these are nanomolar concentrations of this HSP90 inhibitor, you can see that you markedly improve the toxicity of irradiation. And this definitely is combinatorial, because in the absence of radiation, this dose of HSP90 inhibitor isn't doing anything by itself. This effect is time dependent. If you look at the panel on the right, you can see that the longer the pre-exposure to HSP90 inhibitor, the better the sensitizing effect, going from 4 hours pre-exposure, which essentially has no sensitizing effect, to 12 hours pre-exposure, which has a significant effect. You get logs more cell kill. So far, the data suggest, and additional information that has come out since this, support the fact that indeed you can affect the robustness of cancer cells with relatively low dose HSP90 inhibitors. So how do HSP90 inhibitors inhibit HSP90? So it was unclear when we got into this, and we identified the first small molecule inhibitor of HSP90, which is a version of 17-AAG, in 1994. It was unclear whether HSP90 was a chaperone that required ATP or not. There are examples of both kinds of chaperones. Using these small molecule inhibitors, it's become very clear that ATP binding and hydrolysis are essential for HSP90 function, and I'll show you why. As it happens, these first inhibitors of HSP90 that were identified, 17-AG is a derivative of galdanomycin, and then a second structurally distinct inhibitor is called radicicol, and those are both shown on the right. This is the end-domain ATP binding pocket of HSP90, and you can see ATP bound here on the left. And to everybody's surprise, these small molecule inhibitors bind in the ATP binding pocket. So one reason why it was controversial up until this point whether HSP90 bound ATP, required ATP, etc., is that its binding affinity for ATP is extremely poor, and its ATP hydrolyzing activity is extremely poor. But with these drugs, we can now specifically measure HSP90 dependent ATPase activity, because only the ATP bound to HSP90 is going to be affected. So contaminating proteins and preparations like kinases, etc., with much higher ATPase activity are not going to be inhibited by these drugs. And you can see the very small amount of ATPase activity coming from HSP90 is fully inhibited by these agents. So HSP90 exists as a dimer. The dimerization domain is in the C-terminal part of the protein. ATP binds to the N-terminal part, shown in blue. And in the presence of ATP, HSP90 undergoes a series of conformational changes, which is important for ATP hydrolysis and for chaperoning of clients. In the presence of these HSP90 inhibitors, however, ATP can't bind because the inhibitors have much higher affinity for this binding site than does the nucleotide. And HSP90 remains in this inactive conformation, which leads to the degradation of any client proteins bound to HSP90 via the proteasome. And we had identified two E3 ligases that ubiquitinate proteins so that they are recognized and degraded by the proteasome. Both CHIP and COLN5 actually associate with HSP90 and are responsible for ubiquitinating clients that are not allowed to proceed through a chaperone cycle because HSP90 inhibitors are blocking it. So development of HSP90 inhibitors picked up quite dramatically after we reported the first one in 1994. And you can just see here over the last 25 years or so how many HSP90 inhibitors have been developed. And all of the ones shown here have been evaluated in clinical trials. The little red top to the panel on the right-hand side is a special case of an HSP90 inhibitor that I'll get to in the last part of the talk. But suffice it to say that a lot of HSP90 inhibitors have gone into clinical trial, and yet no inhibitor has yet been approved by the FDA as having any single agent activity. And these, unfortunately, are the few HSP90 inhibitors that remain in clinical trial as of August of this year. So nowhere near as many as were shown on the previous slide. And a couple of these trials are simply trials for imaging of HSP90 with iodinated versions of an HSP90 inhibitor. But there still remain a few phase 1 and phase 2 trials that are actively recruiting. These combination trials, which most of these on this table are in combination with previously approved drugs that target this particular kind of cancer. The indication is shown on the right of the slide. And so you can see that now most of the clinical trials are not so much focusing on HSP90 as a single agent, which we would have suggested from the beginning was not the way to develop this drug. It's best used in combination because what the radiation experiment showed a long time ago was that we can affect the robustness of a cell exposed to something else when we put in an HSP90 inhibitor. So a lot of the agents used in combination are kinases, but not all of them are. These trials are open, and it's hopeful that we'll see some interesting results. These are primarily phase 2s. There are some phase 1s also included here. So about 6 years ago, interest was increased that perhaps we could develop an HSP90 inhibitor for clinical use, and in part based on this study in HER2-positive breast cancers, where patients who had been treated with the antibody to HER2, Herceptin, initially respond and then stop responding. And so everybody was progressing on the antibody alone. Now, HER2 turns out to be a kinase, and it's very sensitive to HSP90 inhibitors, very dependent on HSP90. And when these people added in 17-AAG to Herceptin, which, remember, was no longer working in these patients, there was a 63% increase in benefit and a 26% response rate, which is actually quite high. So people got all excited that combination with a HER2 with an antibody that targets HER2 may be a great use for HSP90 inhibitors, and this is still under investigation even now with newer, more synthetic HSP90 inhibitors and with different antibodies to HER2. Some other interesting data that is similar comes from a phase 2 study in non-small cell lung cancer with gantenerumab, which is a more recent HSP90 inhibitor, in patients that are positive for ALK. ALK is a kinase that is frequently mutated in a reasonable percentage of lung cancer patients. And all those green bars you can see that are going down in the presence of gantenerumab as a single agent here shows that patients whose lung cancer expresses ALK mutation seem to be particularly responsive to this HSP90 inhibitor. The yellow bars are lung cancers with RAS mutations. A significant number of them show some response as well. So this is also something that's ongoing. Some anecdotal evidence of single-agent activity of gantenerumab in ALK-positive lung cancer is shown here. Which one of these is the light? So this is circling the tumor before treatment with the HSP90 inhibitor. And you see this patient, first of all, has stage 4 lung cancer, which is very advanced, received crizotinib, which is an ALK inhibitor, for 1 year and progressed on that, and then enrolled in this trial looking at gantenerumab. And you can see that these tumors, which were no longer responsive to an ALK inhibitor, nonetheless are shrinking after 3 doses of the HSP90 inhibitor. So there are trials underway right now combining ALK inhibitors with HSP90 inhibitors as opposed to just looking at HSP90 as a target after the patient stops responding to the ALK inhibitor. Here's another example in a lung cancer with a BRAF mutation. BRAF is also a client of HSP90. This patient received a lot of chemotherapy as well as targeted agents, including a proteasome inhibitor, and at this point was not responding to anything. But after 4 cycles of HSP90 inhibitor, this lesion went from this size to this size. And the duration was for more than a year. So there are definitely hints of activity, even single agent activity. But we're asking these HSP90 inhibitors to now cause regression of tumors that have stopped responding to everything else that's been tried. And after they develop resistance to each of these treatments, they get more and more aggressive. And this is a very high bar for any agent to cross, including an HSP90 inhibitor. This is a triple-negative breast cancer, again, receiving a lot of treatment and showing what happens after 3.5 months, after 4 months of gantenerumab. So there's a lot of anecdotal data suggesting that HSP90 inhibitors should work in cancer. Why haven't any been approved yet? So we started looking at other things that HSP90 inhibitors might do that might be counteractive of their anti-tumor activity. And one of the things that we and others have found is that HSP90 inhibitors seem to potentiate the heat shock response by activating the master transcription factor of the heat shock response, HSF1, which is activated by the same kinds of stresses that HSP90 looks for. And that's because HSP90 itself is a transcriptional target of HSF1, as is another molecular chaperone, HSP70. So what we're showing here in a Western blot in a cell line is that in increasing, whoops, sorry. Increasing concentrations of galvanomycin cause the degradation of ERB2, also known as HER2, which is that kinase that's very sensitive. But at the same time, they cause an increase in HSP70, which is indicative of this induction of the heat shock response mediated by HSF1. And you'll notice it's only the heat-inducible HSP70 in the red box at the bottom, not the constitutively expressed HSC70, whose levels are going up, suggesting this really is a transcriptional effect that's mediated by HSF1. And if you look in patients receiving an HSP90 inhibitor, you can look at HSP70 mRNA induction in lymphocytes of patients, and you can see from the data shown here that there's a correlation that's quite strong between the dose of HSP90 inhibitor given and the increase in HSP70 mRNA. And the higher the dose and the more cycles of treatment, the bigger the increase in HSP70. And this is plotted in the two graphs on the bottom, and you can see that the correlation is quite remarkable. So this isn't just a phenomenon of cell lines in vitro, it's happening in patients as well. A number of years ago, the Lindquist lab published a paper reporting that not only does HSF1 drive genes associated with the stress response, they also have a program of cancer-specific gene expression. And so you might think that if you're using an HSP90 inhibitor, which is upregulating HSF1 in some fashion, or at least upregulating its activity, if this leads to upregulation of this cancer-specific gene expression, you're going to be shooting yourself in the foot in terms of using this as an anti-cancer treatment. This lab also looked at survival with breast cancer. If now, instead of stratifying for HSP90, you stratify for HSF1, and they found again that the high-expressing HSF1 samples in those tumors, the patient's survival was negatively affected. So again, there's a negative effect of high HSF1 in tumors, just like there is of high HSP90. And if you look across a number of cancers, if you just look at gene expression at the mRNA level, the types of cancers are shown on the x-axis, and it doesn't really matter which ones they are. But you can see in the red bars is HSF1 expression, and the green bars is c-MYC expression. c-MYC is generally considered to be a paradigm of a cancer-related transcription factor that's frequently overexpressed in cancer. And you can see that HSF1 is in fact far more overexpressed as a rule than is c-MYC. So cancers need HSF1. If your anti-cancer drug is in any way activating HSF1, that's not going to ultimately be a good thing. So for a long time, the model of how HSP90 is related to HSF1 is that monomers of HSF1 are sequestered by HSP90 until there's a stress. And when there's a stress and you get misfolded proteins, HSP90 comes off of the HSF1, goes to deal with the misfolded proteins. And these monomers of HSF1, which are not active until they trimerize, are allowed to trimerize and become active transcription factors. That's been a model that's been around since the mid-90s. So if you take this and say, okay, what do HSP90 inhibitors do? The speculation was HSP90 inhibitors acts like another cell stress. So there's an HSP90 inhibitor, binds to HSP90, causes release of HSF1, and then the protomers are allowed to trimerize and become active. So this was the model. HSP90 functions as a repressor of HSF1. It functions as a sensor for proteotoxic stress, and release of HSF1 from HSP90 is the key event in induction of the heat shock response that's driven by the transcription factor. So we asked a number of years ago if this actually makes sense given the data that's accumulated over the last 20 years. And the answer is no, it doesn't make sense. First of all, endogenous interactions between wild-type HSP90 and HSF1 are almost impossible to visualize without cross-linking. So that doesn't suggest that HSP90 is sequestering HSF1 in the cytoplasm, or it would be much easier to see these interactions. And a recent study done in vitro with purified proteins, in fact, suggested if you add HSP90 beta, which is one of the two isoforms of HSP90, to HSF1, it promotes the temperature-dependent trimerization and activation of HSF1. So exactly opposite to what the old model had said. So we re-asked the question, can we visualize stable interaction of HSP90 with HSF1? And to do this, we used two well-characterized point mutants of HSP90. The one on the left, D93 in HSP90 alpha and D88 in beta, they can't bind ATP, and so they remain in what's called an open conformation where the endomains are undimerized. This basically is a non-functional HSP90. But it's a conformation that at ground state is the most common in a cell. The mutant on the right, E47A in alpha and E42A in beta, can bind HSP90, and so the endomains come together, but they can't undergo ATP hydrolysis. And so this endomain dimerized conformation essentially is stabilized. As you might expect, since this is a conformation that is on the way from open to ATP hydrolysis, is a very unstable conformation without this mutation. It's transient, and it's very hard to identify in human HSP90. So when we look for interaction of either wild-type HSP90 or these two mutants with HSF1, we found that only in the case of the E47A and, to a lesser extent, E42A with beta, you could see robust interaction of HSF1 shown in the top plot there when you pull down flag-tagged HSP90. So HSF1 is indeed able to bind to that ATP-bound closed trap conformation, whereas we could see no interaction of wild type or of the open mutant. So this got us to thinking a little. If the ATP-dependent closed conformation is a transient state and only infrequently accessed by the chaperone, what does this mean for what HSP90 is doing to HSF1? We next asked if HSP90 inhibitors have any effect on this interaction. And as you can see on the left-hand group of panels there, these three HSP90 inhibitors, which have all been in the clinic, all dissociate HSF1 quite efficiently from HSP90 and very quickly, whereas two other inducers of the heat shock response and two other activators of HSF1, heat shock itself, shown on the right, and a proteasome inhibitor right next to heat shock, don't cause dissociation of HSF1 from HSP90. So what this says is that this ability of N-terminal HSP90 inhibitors to dissociate this complex is unique, but it is not required for activation of HSF1. So that again goes counter to the model that's been around for quite some time. So we did a reporter experiment based on including the promoter region of HSP70 to which HSF1 binds, coupled to luciferase. So we wanted to see the effect of 17-AAG, heat shock, and the combination on HSP70 promoter-dependent luciferase activity. And what you can see here is that 17-AAG in this transiently transfected model has essentially no ability to induce reporter activity. But compared to heat shock alone, 17-AAG has a combinatorial effect when added to heat shock that's quite significant. It's a 6 to 7-fold increase in reporter activity. And the heat shock, the way the heat shock is given is that cells are put at 42 degrees for 30 minutes. And then in this case, we're allowing a period of recovery back at 37 degrees for 4 hours. And the HSP90 inhibitor was added 2 hours before the heat shock. So we did a chromatin immunoprecipitation experiment, otherwise known as CHIP, using antibodies to HSF1 to look at HSF1 binding to DNA. And what the HSF1 is going to be binding to are those same elements as we were looking at in the promoter to HSP70. So these are elements that recruit HSF1 to promoters that are sensitive to its transcriptional activation. And the readout here is we PCR the amount of the promoter that we're able to pull down with antibody to HSF1. And basically, if you just look at the graph on the bottom, you can see that for 2 promoters, the HSP70 promoter and to a lesser extent the HSP90 promoter, if you just heat shock the cells, and that's the no drug group, N is no heat shock, and those numbers are set at 1. You can see that there's an increase in HSF1 binding to the promoters at 5 minutes after heat shock is over, so recovery time of 5 minutes. But by 2 hours, the amount of HSF1 binding goes below baseline and stays there for the remainder of the experiment, which is a recovery of 6 hours at 37. In contrast, if you add the HSP90 inhibitor for 2 hours prior to the heat shock and then do the same recovery experiment, you can see that even in the absence of heat shock, HSF1 bound to the promoters for 70 and 90 are as elevated as they are in the no drug groups after heat shock. So 17-AAG is causing an increase in HSF1 occupancy of promoters in the DNA. And the other thing I would argue from these data, if you look at the 17-AAG group, is that HSF1 binding to these promoter regions is retained for much longer if you add in drug to the heat shock than you see with just heat shock alone. So even at 4 hours, the levels are, at least for HSP70, above the baseline. And this is counter to what we see in the no drug group. So our model is that HSP90 inhibitors actually block HSF1 dissociation from DNA, which is mediated by HSP90 alpha, as sort of a feedback control on turning off the heat shock response. HSP90 inhibitors prevent HSP90 alpha from coming on and pulling the HSF1 off. And so its occupancy on the promoter is therefore much longer and to a higher extent. And when other factors that are important for driving HSF1 transcription are present, you get increased transcription for a significantly longer amount of time. And we think that's what HSP90 inhibitors are doing. Now, as it happens, we've also been working with HSP70, which is also known to interact with HSF1. And we found that an HSP70 inhibitor seems to block this ability of the HSP90 inhibitor to augment the reporter activity you see after heat shock. So the 17-AG plus JG98, which is the HSP90 inhibitor, is shown in purple on the slide compared to 17-AG and heat shock alone, which is the red line. You can see it's almost completely suppressed. The 70 inhibitor suppresses the heat shock response by itself, so it isn't anything specific to 17-AG, and it's probably happening before HSF1 ever gets to the nucleus. And if you do a chip experiment, in this case with JG98, you in fact find data that confirm this hypothesis. So no drug, just heat shock on the left versus JG98 plus heat shock on the right. And you can see that the amount of HSF1 bound to chromatin after heat shock is markedly suppressed. Complete opposite of what an HSP90 inhibitor would do. And in fact, if you combine the two agents, 90 inhibitor and 70 inhibitor, and now we're looking at a Western blot of HSP70 protein, as well as the very sensitive HSP90 client ERB2 or HER2. In the panels on the left, you can see with HSP90 inhibitor alone, reduction in ERB2 expression, but activation of HSP70, increase in HSP70 levels. On the panel on the right, if you include an HSP70 inhibitor with the HSP90 inhibitor, you still get reduction in the HSP90 client HER2, but now you significantly blunt the increase in HSP70 protein consistent with the chip data and the reporter information that I showed you in the previous slides. So we're thinking that a combination of this HSP70 inhibitor, which we've already used in vivo and has anti-tumor activity as a single agent, but hasn't progressed past a few animal experiments yet. But combination of this inhibitor with an HSP90 inhibitor may allow for retention of the HSP90 inhibitor effect on its clients, but prevent this counterintuitive effect on HSF1, which we would very much like to see. The last few minutes, I just want to get back to the newest trial using an HSP90 inhibitor, which was the red top part of that last bar in one of the early slides. This trial has just actually opened here at NCI. There are only a few patients currently involved, but it's based on some very interesting data we got in collaboration with Lee Hellman and Christine Heskey in the pediatric branch at NCI. And that's using a property of HSP90 inhibitors that are true for HSP90 inhibitors as a class. All N-terminal HSP90 inhibitors have the property of being concentrated in tumors compared to normal cells, and for remaining in tumors for a significant amount of time compared to their normal half-life in normal tissues and blood. So they can remain at high levels in tumors, depending on the inhibitor, anywhere from 2 to 4 days after they're completely gone from the circulation. So the folks at a biotech called Cinta decided to use this property of HSP90 inhibitors to deliver a payload, which in general would be a cytotoxic agent that would have significant systemic toxicity or systemic instability. And the first agent they chose to examine was the active metabolite of irinotecan, which is structurally shown on the right, along with the HSP90 inhibitor. Irinotecan is a topoisomerase inhibitor, and it's used to treat. So what it does is it prevents the repair of DNA strand breaks, and is generally used in conjunction with a DNA damaging drug, or in a cancer that's aneuploid and genetically unstable to begin with. It causes a lot of DNA, of the inability to repair the DNA, and a lot of replicative stress. So this agent is coupled to the HSP90 inhibitor through a cleavable linker, and then it's administered to, in our case, animals with Ewing sarcoma, xenografts, and rhabdomyosarcomas. These are pediatric sarcomas that are treated here at NCI. And I'm not going to belabor the point, but the bottom line is there's a dose-dependent, clear, efficacious effect of these conjugates, and the conjugates termed STA8666. So the orange lines, the light blue lines, and the gray lines, compared to irinotecan alone, which is the purple line, and the HSP90 inhibitor alone, which is the red line. So in these models, HSP90 inhibitor alone does almost nothing. Irinotecan always gives you a very dramatic response. This is true in these mice, but it's also true in patients. But invariably, tumors grow back and they become resistant to irinotecan. And that's also shown in these models. However, when mice received 2 to 4 injections of this conjugate of the active metabolite of irinotecan coupled to the HSP90 inhibitor, we could achieve doses, especially in the Ewing's model shown in B above in the slide there, where you could get complete regression of the tumor, and the tumors essentially never came back. And the ones that did come back, if you retreated, they would go away again and stay away for another extended amount of time. Those arrows show how frequently we inject the drug. So the drug is given once a week. And in the case of the Ewing's model, we gave 2 weekly injections, and that was it. And these tumors are quite large if you look at the size numbers on the y-axis. The rhabdomyosarcomas also show a dose-dependent effect, but it's not as durable as the Ewing sarcomas. And if you look at survival, you can see that as well, especially in the overall survival in the panel on the lower right-hand side, which is the Ewing's model, with the highest dose of conjugate, which is achievable in people. There's essentially 90% survival over about 2/3 of a year. And these data actually were instrumental in initiating the clinical trial that's just open. This is another model of Ewing sarcoma. This is a PDX that never saw irinotecan before. And you can see that while irinotecan is effective in this model, you get what look like complete regressions in the purple line. The tumors invariably regrow. But in the case of the conjugate, they never regrow. And in this case, survival at 200 days is 100%. And I can skip that. I can skip that. This is the trial, which is in clinicaltrials.gov. And with that, I'll stop and acknowledge these people in my lab, elsewhere in NCI, and at Cinta for collaborations and for doing the work. And happy to answer any questions. No, HSP90 is probably the chaperone that actually sequesters HSF1 in the cytoplasm. People thought it was HSP90. It's probably HSP70. So the HSP70 inhibitors actually end up destabilizing the HSF1 as protomers, and so they never get to trimerize and get into the nucleus in the first place. In terms of HSP90, yeah, the model is that in the presence of ATP, HSP90 will go through this conformational change, latch on to the HSF1, and then as a consequence of ATP hydrolysis, pull HSF1 off of the DNA. What the HSP90 inhibitors do is they displace ATP from the binding pocket because they have much greater affinity than does ATP. The dimerized HSP90 now opens up again, and it just falls off of HSF1. And so HSF1 is allowed to remain on the DNA for much longer.
Are there any commercially available HSF1 inhibitors?
Yes, there's some sold as such. They may or may not be specific, and they're not particularly potent.
So they're not in the clinic right now?
No, it's likely this HSP70 inhibitor will be in the clinic first as a single agent. And depending on how that looks, it's difficult to do a clinical trial where you're combining two unapproved drugs. So one of those two would have to get approved before we could combine them. Or if there were an HSF1 inhibitor that got through the clinic and was approved, we could use that, but those don't exist. So there's a ways to go, but we have the beginnings of a potential solution to this problem.
That'll do it, Len.
Great.
Thank you.
Thank you.